welcome to my channel. So today we are going to talk about we are going to continue with the digestive system, but today's lesson is specifically for the accessory organs. So we are going to go. Okay, so let's start. So the accessory organs of the digestive tract, there are a lot. So number one, we are going to start with the salivary glands. So the salivary glands are of three pairs, and these are the parotid gland, the sublingual gland, and the submandibular gland. So specifically in the anatomy, we're talking about this. The parotid gland is this part where the the pointer is, and you have the submandibular gland, and you have the sublingual gland. So these three are the salivary gland. So this is also another picture just to show the sublingual, the submandibular, and the parotid gland. Okay, so let's now start talking about them in details. So the parotid gland is the largest and the most important salivary gland. It's entirely serous gland. It's located below the ear pinna. Okay, and the duct of the parotid gland is also known as Stenson's duct. That's about the parotid. Okay, then you have the sublingual gland. So the sublingual gland is the smallest salivary gland located below or on the base of the tongue, and it opens by means of several small ducts called the ducts of Livinus. Those are the sublingual glands. Okay, then the submandibular gland are one pair and present inside the lower edge of the mandible and drain their secretions through Wharton's duct, and they contribute maximum of 70% of volume of the saliva. Those are the submandibular gland. So now let's talk about saliva. So saliva is in terms of composition, saliva contains 99.5% of water, ions like bicarbonate, sodium, potassium, chloride, and of course etc. etc. And of course it also has enzymes such as ptyalin, lingolipase, antibacterial substances like the IgA, also as lysosome, thiothionates. That's about the saliva. Then in terms of pH of the human saliva is 6.8. Then in terms of functions of the saliva, it binds the masticated food into a bolus for swallowing, and it initiates fat digestion by the lingolipase. It also initiates starch digestion by the alpha amylase. So if you remember very carefully when in our last lecture we were talking about chemical digestion, we mentioned to say in the saliva there is amylase which initiates the starch digestion or the chemical digestion with the enzyme. Okay, then the other thing is, since saliva contains IgA and lysosomes and thiothionates, the other function is to kill the bacteria due to these contents, IgA, lysosome, and thiothionates. Okay, then the other function for saliva is lubrication, so it's also used for lubrication. Okay, then we are done with saliva. Now let's talk about the biliary tree, the biliary system. So the biliary system consists of the liver, the gallbladder, and the bile ducts that are involved in the production, storage, and transportation of bile. Then bile is secreted by the liver cells at a constant rate of about 40 ml per hour. Then when digestion is not taking place, the bile is stored and concentrated in the gallbladder. Then later it is delivered to the duodenum. So when you are asked to draw the biliary system or the biliary tree, this is what you are supposed to draw. You're supposed to draw the liver, supposed to draw the gallbladder, and the ducts of the right hepatic duct and the left hepatic duct. Then where the right and left hepatic ducts join together, it's the common hepatic duct. Then the gallbladder has a cystic duct. Then where the cystic duct and the common hepatic duct join is called the common bile duct. Then finally of the pancreatic duct and the common bile duct join together in the ampulla of Vata. Okay, so this is what we are talking about. This is what you are supposed to draw. So I think I've already mentioned most of this. So in the after the ampulla of Vata, then we're going to find what we call the sphincter of OG. Okay, then finally the duodenum itself. So this is the biliary tree. Okay. So let's start with the liver. So the liver is the largest visceral organ and gland of the body and weigh about 1.2 to 1.5 kg in an adult human. Then it is situated in the right hypochondrium, and the liver cells are called hepatocytes, and they have a maximum regenerative capacity. Then the Grison's capsule, which is of course the capsule of the liver, that's the capsule of the liver is called the Grison's capsule. Then the liver is divided into right and left borders by a ligament, which is known as the falciform ligament, which is just a fold of peritoneum. Then you have also right lobe forms, and the right lobe forms fifth out of sixth over the liver, and the liver also has four lobes. Okay, so they have the right lobe, left lobe, the caudate lobe, and the quadrate lobe. So the quadrate lobe is here, then the right lobe, left lobe, and the caudate lobe this side. Okay, so this is what we are talking about. The right lobe, left lobe, and the caudate, and the quadrate. Quadrate, yeah, quadrate lobe. Okay. Then we have another thing just deep and the transverse fissures situated between the caudate and the quadrate lobe. We call it the porta hepatis. So the contents of the porta hepatis are the portal vein, hepatic artery, and the common bile duct. That's the porta hepatis. Okay, then in terms of blood supply for the liver receives 20% of its blood supply through the hepatic artery and 20% through the portal vein in terms of drainage. Then functions of the liver. Number one, metabolism of carbohydrates, fats, and proteins. The other function is to synthesize bile, which help in fat digestion and the prothrombin. Then also excretes drugs, toxins, poisons. So an example of poisons, of course, alcohol is one of the poisons we're talking about. Then of cholesterol, bile pigments, and heavy metals. And the other function is hematopoiesis. So the liver is the most important site of red blood cell production in embryonic. Then the fifth function is synthesis of the plasma proteins, albumin, globulins. Then all plasma proteins except immunoglobulins are produced by the liver. And the albumin is very important because we find that if you are to learn most conditions such as liver cirrhosis, where there is now depletion of albumin, so someone will develop hypoalbuminemia, so meaning there will be low levels of albumin, which will cause conditions such as ascites and edema, of course. Those are some of the complications that may come in if the liver was damaged and it stopped producing albumin. Okay, so let's continue. So sixth function, it's also important for storage of glycogen, iron, fat, and vitamin A and D. It also produces urea from ammonia, which is the waste product of protein metabolism by the ornithine cycle. It produces urea from ammonia. Okay, then the other function is in the Cori cycle, there is conversion of lactate into glucose. So this is also another function. Then the other one is secretion of heparin, which is an anticoagulant. Then, of course, the liver has another function of destruction of bacteria, but this one is done by the Kupffer cells, which are the macrophages of the liver. Okay, so then in terms of metabolism of carbohydrates. So carbohydrates can be metabolized in different forms. So number one, we can metabolize through the process of glycogenesis. So in the liver, excess glucose are withdrawn from the portal blood by the liver cells and converted into glycogen granules for storage. Okay, then if of course when during the process of glycogenolysis, this occurs when normal amount of glucose in blood drops, then the liver cells convert glycogen into glucose and release the same glucose inside the blood. So the one for glycogenesis is instituted by hormones. Even in glycogenolysis is also instituted by hormones. Okay, so but specifically in terms of glucagon is one which stimulates the conversion of glucose to glycogen. Okay, so let's make progress. So then there's also another metabolism which is known as gluconeogenesis. So at the time of need, liver cells convert non-carbohydrate compounds such as amino acids, fat acid, and glycerol into glucose. So as the name suggests, gluco for glucose, then neogenesis, so meaning you are making something new. Okay, you are building. Okay, so gluconeogenesis is, of course, making glucose from non-carbohydrate precursors. Okay, so let's make progress. So we are done with the liver. Let's go to the bowel. So the bowel, of course, is formed in the liver, and the human liver secretes about 0.5 to 1 liter of bile per day. It is a greenish yellow fluid formed in the liver and stored in the gallbladder, and it's composed of 98% water, bile salts, bile pigment, cholesterol, and inorganic salts and iron. Then functions of bile: it's important for absorption and emulsification of fat. It's important for absorption of soluble fat soluble vitamins. So the fat soluble vitamins, of course, is A, D, E, K. So these are fat soluble vitamins. Okay. And then it's also responsible for activation of pancreatic enzymes, such as, so the bile salts activate the pancreatic lipase, to be specific. Okay, so that's about bile. So now let's go to the gallbladder. So the gallbladder is a pear-shaped reservoir of bile situated in the fossa on the inferior surface uh, of the right lobe of the liver. Okay, that's the gallbladder. Okay. The functions of the gallbladder is storage of bile. So it has stores about 30 to 50 mils capacity. It has 50 to 50 mils capacity, and it's released into the duodenum when required. Then it's also responsible for absorption of water and concentration of. Okay, then in terms of the other structures found in the biliary tree, you are going to find the cystic duct, which is the duct of the gallbladder, and it joins with the common hepatic duct to form the common bile duct. And the bile duct joins with pancreatic duct to form the hepatopancreatic duct, which opens at the ampulla of Vata. So the common bile duct and the pancreatic duct are going to join together at the ampulla of Vata. Okay, so this is the structure you are talking about. So let's make progress. So the common bile duct and the pancreatic duct going to join together in the ampulla of Vata, then finally into our body, then into the duodenum. Okay, so now for the pancreas, I think we can now talk about the pancreas. So this is how the pancreas looks like. It has the tail. This is the tail. Then the one which is near the duodenum is the head. Okay. So this is what we are talking about. To be specific, it has the head, it has the body, and it has the tail. So this is the pancreas. Okay, and it looks like a leaf actually. So the pancreas is an endodermally derived mixed gland, or it's an heterocrine gland, or a compound gland. It is made up of 99% exocrine and 1% endocrine part. So meaning the pancreas is actually both exocrine and endocrine organ. Okay. Then it's made up. It also secretes what we call pancreatic juice. which is a colorless, highly alkaline, about a pH of 7.5 to 8.3, and contains about 96% water and 4% salts and enzymes, and is a complete digestive juice containing digestive enzymes for carbohydrates, proteins, and fats. Then the same pancreatic enzymes are: one, we have proteases. So for protease, we have trypsin, chymotrypsin, and the carboxypeptidase. So they convert proteins to amino acids. So protein, then if you want to convert to an enzyme, protease. Amylase is responsible for starch and the glycogen, so it converts these to maltose. Then the enzyme that converts maltose to maltose is the maltase enzyme, but specifically for amylase is one which initiates this mechanism. Then the lipases converts complex fats to fat acid and glycerol. Then I think we are done on there. Portohepatis. So the portohepatis, or the hepatic portal system, is this one we are talking about. We're talking about the blood vessels and of course other. So in terms of portal circulation, we talk about the hepatic portal vein, which carries about 75 to 80% of the hepatic blood flow, and hence is crucial. Then it is not a true vein, as it does not conduct blood directly to the heart. It supplies veins with metabolic substrates. It also ensures that food ingested is processed by the liver first before entering the systemic circulation. This way, ingested toxins are detoxified by hepatocytes. It also ensures that the liver is not the first organ to absorb nutrients after the intestine. Then that's the end of the lecture. If you haven't subscribed, please subscribe and hit the bell notification. And we have last lesson for the digestive system. So we started with the digestive system in terms of anatomy, talk about the accessory organs. So next time we're going to talk about the peritoneum. So then that will be all on the digestive system. Then it means we are done with the digestive system. Okay, so see you on the next tutorial video, which is on the peritoneum. Okay, so we talk about the anatomy, structure of the peritoneum, and of course as well as the functions of the peritoneum. All right, thank you.